By every rule of population genetics, the signal should have disappeared, not weakened, not reduced. Disappeared. 3,000 years is a very long time for any population to maintain a recognizable genetic signature. Entire civilizations have emerged and vanished in less time. Empires have risen, conquered continents, collapsed, and been forgotten. Languages have been born, evolved, and died. Borders have moved thousands of times. And yet, when scientists began comparing Jewish communities scattered across multiple continents, they found something that seemed almost impossible. The same genetic signal was still there. Not in one isolated village, not in one country, not in one branch of the population. Across communities separated by oceans, deserts, mountain ranges, and more than 1,000 years of independent history. How was that possible? To understand why researchers were so surprised, you first need to understand what they expected to find. Because history suggested a very different outcome. Imagine taking a population and scattering it across the world, not for a few decades, not for a few centuries, for nearly 2,000 years. Some groups settle in Eastern Europe, others move into North Africa, others establish communities across the Middle East, some eventually reach Ethiopia. Each community develops unique customs, unique accents, unique daily experiences, different foods, different clothing, different political realities. Generation after generation passes. The original homeland becomes more distant. The expectation in population genetics is straightforward. Over time, local influences accumulate, host populations are larger, intermarriage occurs, migration continues. Genetic drift begins to reshape the population. Eventually, the original signal becomes difficult to detect. This pattern appears throughout human history. In fact, it is so common that many researchers assumed something similar would happen here. But before we get to what scientists discovered, we need to understand how this extraordinary journey began. The story starts in the southern Levant, a region connecting Africa, Asia, and the Mediterranean world. For thousands of years, this narrow corridor served as one of the most important crossroads in human history. Trade routes passed through it, empires fought over it, cultures mixed within it. Ancient kingdoms rose and fell across its landscape. Long before modern nations existed, people were farming, building cities, establishing trade networks, and creating communities throughout the region. Archaeologists have spent decades excavating these ancient sites. Beneath layers of soil, they uncovered homes, pottery, inscriptions, tools, burial grounds, and evidence of societies that existed thousands of years ago. For most of history, those discoveries told us only part of the story. Artifacts can reveal how people lived. They can reveal what people built. They can reveal where people traded, but they cannot always reveal biological continuity across thousands of years. DNA changed that. Yet long before genetic science entered the picture, history introduced a challenge that would shape everything that followed. The challenge was dispersal. In the 1st century CE, the political landscape of the region changed dramatically. Conflict with the Roman Empire culminated in one of the most consequential events in Jewish history, the destruction of the Second Temple in Jerusalem. What followed was not a single migration, it was a long process. Communities spread outward over generations. Some moved east, others moved west. Some settled in regions where Jewish communities already existed. Others established entirely new centers of life far from the original homeland. By the medieval period, Jewish communities could be found across enormous geographic distances, from the Iberian Peninsula to Persia, from North Africa to Eastern Europe. From major trading cities to isolated settlements, the scale of dispersal was extraordinary, and that scale is exactly why scientists became interested. Because every additional century should have increased genetic divergence. Every new host population should have introduced additional variation. Every migration should have pushed communities further apart. Yet historical records hinted at something unusual. Despite geographic separation, these communities maintained strong cultural connections. Religious texts remained central. Shared traditions continued. Common legal frameworks persisted. Communication networks connected distant communities. A Jewish merchant in one region could often interact with a Jewish community hundreds or even thousands of miles away and find familiar customs, familiar practices, and a recognizable shared identity. Historians had documented this phenomenon for generations. The question geneticists wanted to answer was different. Could that cultural continuity leave a biological signature, or would 2,000 years of separation erase it completely? The only way to find out was to test it. And by the late 20th century, scientists finally had the tools to try. Advances in genetic sequencing were transforming population studies. Researchers could now examine specific markers passed down through generations. Some markers followed paternal lines, others followed maternal lines. Still others allowed scientists to compare entire populations and estimate how closely related they remained after centuries of separation. The methodology was becoming increasingly sophisticated. The datasets were growing larger. And one group of researchers decided to tackle a question many had considered impossible. What happens when you compare Jewish communities from around the world and measure their genetic relationships directly, not through tradition, not through historical records, not through religious texts, through DNA itself? The project would eventually involve thousands of individuals representing communities spread across multiple continents. Ashkenazi communities from Eastern Europe, Sephardic communities connected to the Iberian diaspora, Mizrahi communities from Iraq, Iran, and surrounding regions, Yemenite communities, North African communities, and others. The expectation seemed reasonable. Researchers anticipated finding significant differences. After all, these populations had experienced centuries of separate histories, different environments, different neighboring populations, different migration paths. Many scientists assumed the shared signal, if it existed at all, would be faint, perhaps barely detectable. Instead, the first results created an entirely new mystery. The communities were different, but not nearly as different as they should have been. And the more researchers examined the data, the stranger the pattern became. Because what they were seeing suggested that something had survived thousands of years of migration far better than anyone expected. The question was no longer whether a signal existed. The question was how it had managed to survive at all. And to answer that, scientists would need to compare Jewish DNA not only with modern populations, but with every major theory that attempted to explain where that signal came from in the first place. The first results created a problem, not because they were unclear, because they were too clear. Researchers had expected Jewish communities scattered across different continents to show substantial genetic divergence. That would have been the normal outcome. After all, these populations had spent centuries living in different environments and alongside different neighboring groups. The expectation was separation. What the data showed was connection, not perfect connection, not identical genetics, but enough similarity to force scientists to ask a new question. What exactly were they looking at? To answer that question, researchers began examining specific genetic markers that can persist across generations. Some markers follow paternal ancestry through the Y chromosome. Others trace maternal ancestry through mitochondrial DNA. Still others compare entire genomes to identify shared ancestry patterns across large populations. Think of it like a massive historical puzzle. Every genetic marker is a tiny clue. One clue means very little. Thousands of clues begin to reveal patterns, and patterns are what geneticists care about most. Because genetics rarely tells stories about individuals, it tells stories about populations. As larger datasets became available, researchers began comparing Jewish communities with surrounding populations. This was where things became interesting. Ashkenazi Jewish communities in Europe clustered together genetically. That was not surprising, but they also showed connections to other Jewish populations outside Europe. Sephardic communities displayed similar patterns. So did many Mizrahi communities. Despite centuries of geographic separation, researchers repeatedly found evidence suggesting shared ancestral origins. The details varied, the proportions varied, the histories certainly varied, but beneath those differences, scientists kept finding signs of continuity. For some researchers, the findings raised an uncomfortable possibility. Maybe several popular theories were incomplete, because for decades, competing explanations had attempted to explain the origins of modern Jewish populations. Some theories argued that Jewish communities primarily descended from local populations that converted over time. Others suggested that later migrations had completely replaced earlier ancestry. Still others claimed that any ancient genetic connection would have been erased long ago. The new evidence forced those assumptions back onto the table. One of the most debated examples involved the Khazar hypothesis. The theory proposed that a significant portion of Ashkenazi Jewish ancestry originated from the Khazar Khaganate, a medieval kingdom located north of the Caucasus. Historical records suggest that elements of the Khazar elite may have adopted Judaism. From that fact, some writers proposed a broader theory. What if large numbers of modern Ashkenazi Jews descended primarily from Khazar populations rather than earlier Jewish communities? It was an idea that generated enormous attention. And for years, the debate remained unresolved. Historical evidence alone could not settle it. This is where genetics entered the discussion. Researchers began testing whether the expected genetic signatures appeared in the data. If the Khazar hypothesis explained most Ashkenazi ancestry, scientists would expect to see strong clustering patterns pointing primarily toward populations associated with that region. Instead, the results were more complicated. The data did not support a simple replacement model. While Jewish populations clearly showed evidence of admixture and interaction with neighboring groups over centuries, researchers continued finding ancestral components linking multiple Jewish communities to one another. The picture emerging from genetics was not one of complete replacement. It was one of continuity combined with mixture. That distinction mattered because human populations are almost never genetically pure. Migration is normal, intermarriage is normal, cultural exchange is normal. The question was never whether mixture occurred. The question was whether an older signal remained detectable beneath it. And increasingly, the answer appeared to be yes. The Khazar debate was not the only theory facing scrutiny. Another explanation suggested that European Jewish populations might be overwhelmingly European in origin. Again, researchers tested the idea directly. And again, the results proved more nuanced than either side expected. Jewish populations often showed evidence of European ancestry. That was hardly surprising after centuries of residence in Europe, but they also consistently displayed genetic components that could not be explained solely through recent European origins. The deeper scientists looked, the more they encountered the same recurring pattern: connection, not uniformity. Connection across regions, across centuries, across populations. Then came another challenge. Some critics argued that any similarities could simply reflect broad Middle Eastern ancestry rather than continuity from ancient Jewish populations. That possibility also required testing, because the Middle East has always been a crossroads of migration. People moved constantly. Empires expanded and contracted. Trade routes connected distant regions. Genetic overlap was inevitable. Researchers therefore needed a more precise method. They needed a way to compare modern populations directly with people who had actually lived in the ancient Levant. For years, that seemed impossible. Ancient populations do not leave behind DNA records waiting in archives. They leave bones, burial sites, fragments. And until recently, extracting usable genetic information from ancient remains was extraordinarily difficult, but technology was improving rapidly. Ancient DNA research was entering a new era. Laboratories developed methods capable of recovering genetic material from remains thousands of years old. For the first time, scientists could move beyond theory. Instead of guessing what ancient populations looked like genetically, they could test it directly. The implications were enormous. No longer would researchers need to rely entirely on modern populations as proxies for the past. They could compare ancient genomes with modern genomes, directly, scientifically, empirically, and that possibility changed everything, because now the question was no longer which modern theory sounds most convincing. The question became, what happens when ancient DNA enters the conversation? Researchers knew the answer could potentially overturn decades of assumptions. It could support existing theories. It could challenge them, or it could reveal something nobody expected. As ancient samples from the Levant became available, geneticists prepared for one of the most important comparisons ever conducted in this field. If the shared signal truly reflected ancient continuity, evidence should appear in those comparisons. If it did not, many existing interpretations would need to be reconsidered. The stakes were suddenly much higher, because this was no longer a debate between competing historical narratives. It was becoming a direct comparison between the ancient past and the living present. And when that comparison finally happened, researchers discovered something that transformed the entire discussion. The genetic signal had not merely survived. It appeared to reach much further back than many people imagined. The mystery was about to enter its most important chapter. For decades, the debate continued because researchers lacked one critical piece of evidence: the ancient population itself. Modern DNA can reveal relationships. It can show patterns. It can identify connections, but there is always a limitation. You are comparing living people to other living people. The deeper question remains unanswered. How closely do those populations resemble the people who lived thousands of years ago? For most of human history, there was no way to know. Then ancient DNA changed everything. Advances in genetic sequencing allowed scientists to recover usable DNA from human remains preserved for thousands of years. Suddenly, researchers were no longer limited to modern comparisons. They could compare living populations directly to ancient populations, and that possibility transformed the investigation. Archaeological excavations throughout the southern Levant had uncovered burial sites dating back thousands of years. Some remains belonged to populations that lived during the Bronze Age. Others came from the Iron Age, the period associated with the emergence of ancient Israelite society. The DNA extracted from these remains represented something extraordinary, a direct biological snapshot of people who had lived in the region roughly 3,000 years ago. Now the test could finally happen. Scientists compared ancient Levantan DNA with the genetic profiles of modern Jewish communities from around the world. If the continuity hypothesis was wrong, the connection should be weak. If centuries of migration and admixture had erased the original signal, researchers would see little resemblance. But that is not what happened. The comparison produced one of the clearest patterns in the entire investigation. Ancient Levantan genetic signatures appeared repeatedly across modern Jewish populations. Not just in one community, not just in one country, across multiple branches of the diaspora. The exact percentages varied, the amount of admixture varied, the historical experiences varied, yet the underlying signal remained. For scientists, this was the moment the mystery began to resolve. The question was no longer whether continuity existed. The evidence suggested it did. The question became how that continuity managed to survive, because from a purely geographic perspective, it should not have. Most populations maintain genetic continuity through isolation. Mountains, deserts, remote islands, physical barriers reduce interaction with surrounding populations, but Jewish communities rarely enjoyed those advantages. For most of the last 2,000 years, they lived within larger societies, inside empires, inside kingdoms, inside cities, surrounded by populations far larger than themselves. Under those conditions, genetic absorption is usually expected. Yet the data suggested something different happened. The answer was not geography, the answer was culture. Researchers increasingly concluded that one factor mattered more than any other: endogamy, the practice of marrying within the community. Across centuries and across continents, Jewish communities maintained social and religious structures that encouraged marriage within the group. This was not unique in human history. Many communities have practiced forms of endogamy. What made this case remarkable was the scale. The practice persisted across enormous geographic distances and over extraordinary periods of time. A community in North Africa, a community in Persia, a community in Europe, separated by thousands of miles, yet often operating under similar cultural frameworks. Shared traditions reinforced shared identity. Shared texts reinforced continuity. Shared religious practices created a sense of belonging that transcended geography. The result was measurable. The genetic signal remained surprisingly stable, not unchanged. No population remains unchanged for 3,000 years, but stable enough to remain detectable, stable enough for scientists to identify, stable enough to connect modern populations with ancient ones. And that finding carries an important lesson, because it reveals something broader than Jewish history alone. It demonstrates that cultural systems can influence biological continuity in powerful ways. People often imagine genetics and culture as separate forces. In reality, they frequently interact. Culture shapes behavior. Behavior influences population structure. Population structure influences genetic patterns. The story researchers uncovered was not simply a story about DNA. It was a story about the relationship between biology and culture across thousands of years. Yet there is an equally important point that often gets lost in public discussions. What genetics can show is not the same as what genetics can prove. DNA can identify ancestry. DNA can reveal relationships. DNA can estimate historical connections between populations. But genetics cannot determine personal worth. It cannot determine identity. It cannot determine political rights. It cannot determine moral authority. Those are different questions entirely. Scientists themselves repeatedly emphasize this distinction. Population genetics describes biological history. It does not decide contemporary debates. And understanding that limitation is just as important as understanding the discoveries themselves. Because the most remarkable part of this story is not what the DNA proves. The most remarkable part is what survived. Think about the scale of the journey: 3,000 years, empires rising and falling, wars, exile, migration, persecution, cultural change, linguistic change, economic change, political change. Nearly every force that normally fragments populations over time. And yet a recognizable ancestral signal remained. Not perfectly, not completely, but undeniably. For researchers, that survival became the real story. The signal traveled from the ancient Levant to Baghdad, from Baghdad to North Africa, from North Africa to Spain. From Spain to Eastern Europe, from the Middle East to Ethiopia, across continents, across centuries, across some of the most dramatic historical transformations in human history, and still traces remained. Perhaps that is why these studies attracted so much attention, not because they solved every mystery, they didn't. Not because they ended every debate, they haven't, but because they demonstrated something many people considered unlikely. A dispersed population can preserve continuity across extraordinary spans of time. The signal survived, not because geography protected it, not because history was easy, but because generations of people carried it forward. One family at a time, one community at a time, one century at a time, and that may be the most important lesson of all. When scientists began this investigation, they were searching for ancestry. What they ultimately discovered was endurance, a genetic signature that crossed continents, a cultural framework that outlasted empires, a connection between ancient and modern populations that remained visible after 3,000 years. The remarkable part is not that researchers found the signal. The remarkable part is that it survived at all, through exile, through migration, through dispersion, through history itself. And perhaps that tells us something important about how human communities preserve themselves across time. If you enjoyed this documentary, subscribe for more evidence-based explorations of history, ancestry, archaeology, and the discoveries changing what we know about the past. Because some of history's most fascinating stories are still hidden in the evidence, waiting to be found.